what we know about it, that medicinal plants are basically uh, dependency of mankind on natural resources for therapeutical purposes since very ancient time, with the development of civilization, and there are over more than 35,000 medicinal plants all over the world, and in India also more than 2,500 medicinal plants are practiced in different type of medicinal systems, and 90% of these plants are harvested from basically from its natural resources, from wild resources. So according to WHO, World Health Organization, around 80% of the developing population is dependent upon the medicinal herbs for primary healthcare needs. So overall, 25% of the prescribed medicine developed in the developing countries as well as developed countries, they are derived from plant-based species. So actually, it is not only the traditional. It is the medicinal plants are not only practiced by traditional, as well as it is also practiced by modern uh, medicinal systems also, like allopath. Next, please. So in India, India is a home of great biodiversity, and India is a part of basically 4 global biodiversity hotspots of the world. So actually, hotspot is what is hotspot? Biodiversity hotspot. It is a site where a huge number of biodiversity is available in the form of endemic species, and in India there are 4 biodiversity hotspots. However, all over the world there are around 36 biodiversity hotspots. So you can see that how rich the biodiversity of India is. So the dependency of natural resources is in our cultural system. Traditional medicinal folklore of the in the form of Ayurveda, Unani, Siddha, and different kind of medicinal systems are widely practiced in India in different forms, in different societies also. So more than 2,500 of species of the medicinal plants are used all over the country, and more than 100 species are used as medicinal in the modern system of medicines also, that is allopath. So you can say that this industry is not only reached around 80 billions in 2020, but it has a great pace of annual growth. It is around 50% currently, and it is expected that it will be reach around 20% growth in next few years. And next, please. And these medicinal plants are the source of great drug discoveries also in the world. You can that in last 40 years, around 1900 drugs were approved by USDA, and around 25% of these drugs are originated from plant-based product, even in semi-synthetic or fully natural molecules. And similarly, around 47% of the totally 155 clinically approved anti-cancerous drugs between the period of 1981 to 2006 was derived from natural product, and it was around 99% from medicinal plants. So half of the anti-cancerous drugs are originated from medicinal plants. And not only in India, but in developed countries like US also, around 118 prescriptions out of 150 prescriptions are based on natural product. So you can judge that most of the discoveries that are currently going on, it is based on natural products. It is based on plant-based molecules only. Next, please. So with this high great demand of medicinal plants, there is a risk of extinction of some medicinal plants also. So the medicinal plant industry is increasing with the annual growth of 15% globally, and species in their natural habitat have the threat of extinction due to overexploitation, overgrazing, or current climatic changes, also climate uncertainties like changes. So the depletion of the natural population of medicinal plants is continuously decreasing in the form of raw material availability also. Around 1500 species of the medicinal plants are going to be extinct in next few years. Destruction as well as unregulated unscientific exploration. So approximately 20% of wild resources have already been exhausted due to human population and plant consumption. Next, please. So due to this high pressure on medicinal plant in the nature, some conservation techniques has been developed for their long-term sustainable utilization. So among the different kind of conservation techniques, these are categorized into two kind of methods: ex situ conservation and in situ conservation. Ex situ means on-site conservation, and in situ means in situation or inside conservation, and ex situ means outside their natural populations. So if we talk about the role of biotechnology in conservation of medicinal plants, I have actually compiled all the information in 3 different sections, followed by further some few sections also. So we will discuss these sections as tissue culture for mass multiplication for commercial purposes, as well as for habitat enrichment and secondary metabolite production. Similarly, I have compiled information related to genetic diversity assessment under conservation priorities development. It's evolutionary studies for ecological processes as analysis, as well as genetic resources for variety improvement analysis. Similarly, in third section, I have compiled information under drug discovery under the heading of characterization of novel molecule and chemical synthesis, chemical or semi-synthesis of active molecules. Next, please. So, what is tissue culture? Generally, you have studied in your class also that tissue culture is a technique of growing plant cells, tissue, or organs, seed, or any part of plant in sterile medium, in a nutrient medium, under the controlled environmental conditions, growth conditions. So, tissue culture is, next please. There are few applications of tissue culture. I think that you already know about it in your class also, that through tissue culture we can do the mass multiplication of plants for production of several thousand plants from a single clone. Suppose there is a need of large number of plants that is not available in such quantity. So tissue culture is the best option for production of large number of plants. Similarly, through tissue culture, we obtain the 2 type clones for quality maintenance. If we have identified any high yielding variety or high yielding plant, high yielding genotype from nature or through any experiment, then we can only multiply this kind of plants through tissue culture techniques. Similarly, for year round of production and to avoid the seasonality, if seeds and propagating materials is not available in other seasons, so the tissue culture remains the only method for supply to pharma or any year round consumption methods. Similarly, production for mature plants actually it reduces the flowering time and fruiting time also. As you know that in some species, some specific phenological events like flowering, fruiting, it is controlled by age. And we cannot wait for flowering or fruiting for more than 5, 6, or 10 years. If there is a demand of any industries that give us some plants immediately, reproductive parts also immediately, then tissue culture remains the only solution. So fortunately in tissue culture, the age of mother plant is added to the tissue culture raised plant. So the flowering, fruiting, or this kind of phenological event, we can get early fruiting and flowering type of things. Similarly, to produce the disease or virus resistance free plant, we can generate plant or pathogen free plant through the tissue culture techniques for any through the convention through the that is not possible through the any conventional propagating techniques. Similarly, for long-term germplasm storage through of tissue culture, raised plant in tissue banks. Through this technique, we can store plant in low nutrient medium solutions for low and under lower temperature for a long time. Also, to solve the problem of seed germination, if seed germination is a issue of seed germination, sometimes maybe a issue for many plants, and if the seed supply is not so enough, then the tissue culture remains the only viable option for production of plant. Furthermore, production of fully genetically transformed plant in genetic modification experiments or any techniques, we can only transform few cells, and through tissue culture techniques, we can then propagate these cells into full plants. Similarly, for production of secondary metabolite in various plants obtained through the tissue culture techniques are richer in medicinally important secondary metabolites. So use of elicitors can be applied for further enhancement of such medicinally important phytochemicals or secondary metabolites. Next slide, please. So what is the principle of tissue culture? Actually, tissue culture of plant is dependent upon the two major characters of cells, that is totipotency and plasticity. What is totipotency? Actually, totipotency is the ability of a plant to regenerate into a whole plant, that is called totipotency. And meristematic containing tissue have such property to regenerate into a new complete plants. Similarly, plasticity is another characteristic that we need in tissue culture. Actually, plasticity is the ability of plants to alter their metabolic growth and any other developing activities according to their environment. So their adaptation is utilized for production of different tissues. So meristematic tissue have such ability to alter the metabolism according to the environment. Therefore, meristematic containing tissue like you can say cotyledon, leaf, ovary, protoplast, root, anther, and any plant, any plant or organ that contain meristematic tissue can be used for generation of new plant. Next slide, please. So what is the basic requirement of tissue culture? Basically, six basic categories of requirements have been described for tissue culture. These requirements are appropriate explant, growth medium, aseptic conditions, growth regulator, and light humidity, that is environment. So for appropriate explant, tissue culture is initiated through the multicellular tissue that is called explant. So explant may be a wide range of tissues that contain meristematic tissue. It may be leaf, root, petiole, cotyledon, embryo, or any other meristematic containing body of plant. Similarly, media consists of all the nutrients that plant required in the form of salts and carbon sources, inert supportive matrices also. Explant. So the explant is after sterilization, explant is placed in the solid surface medium, and then sometime it can be directly placed into the liquid medium also. You can see in photographs that sometime liquid medium is used. Similarly, aseptic conditions is very important because there are always chance of attack of bacteria, fungus, or other microorganisms. So sterilization is required of the tissue culture experiment to remove all the contaminations. So the different kind of sterilization techniques are used in the tissue culture, like chemical treatment or sterilization of explant or any material that we use, any equipment we used. Similarly, surface sterilization of explant is completed through the uh, different means, like use of some solvents as well as some conditions. Plastiwares and autoclave is used for autoclave is used for the sterilization of chemicals, media, glasswares, handling tools, etc. And sometimes dry treatment is also used for dry heat treatment is used for handling tools, small equipment, and metallic elements, metallic things, equipments, etc. And growth regulators are also used in the form of oxygen and cytokines. Sometimes other growth of, regulators are also used, but oxygen and cytokines are major important growth regulators. Similarly, the environmental conditions like light, humidity, and temperature are also required to maintain for proper growth of eggplant in controlled environment. Next, please. Next slide. Yeah. So the culture medium. Actually, different kind of culture mediums are available in the market, and due to their different media compositions, they are categorized into different types. So different commercially available medium have the different salt concentration, and they have different even presence and absence of these salts also as per the requirement of plant. Some preferred mediums of the tissue culture experiments are N5 medium, BKW medium, GB5 medium, Camberg medium, etc. So you are in your classroom studies, you have also, I think you already know some kind of media also. But the most commonly used media is MS media, that is that was discovered by Morosky and Skog in 1962. So the uh, different micronutrients carbon sources, vitamins, organic salts, and growth regulators are used in this mediums. Major elements are nitrogen, phosphorus, potassium, sodium, calcium, iron, and magnesium as in macronutrients. So in macronutrients, you can see that nitrogen is a part of nucleic acid, that is DNA, proteins, chlorophyll, amino acids, and hormones. So it actually basically it influences the growth of plant directly. So it is a very essential element. Similarly, phosphorus is a part of nucleic acid, DNA, phosphorylation. It is also used in the phosphorylation of DNA during gene expression. It is abundantly used in meristematic and fast-growing tissues for essential photosynthesis, respiration, and many other important biological properties, physiological processes. Potassium, it is required for maintaining the growth of cell, for ionic potential, for cellular transport, etc. Similarly, calcium, it works as a secondary messenger in the plant cells, and it is also involved in the formation of cell wall, root, leaf development, particularly in it participates in the translocation of sugar, amino acid, and many other molecules like oxalic acids as toxins. And iron, you know that it is involved in the respiration, the chlorophyll synthesis, and in the form of sodium FeNAEDTA as EDTA, sequester, iron, and many other available forms. Similarly, magnesium, it is involved in the photosynthesis respiration system, and it actively participates in the uptake of phosphate and translocation of phosphate starch and many molecules in the xylem and phloem. Similarly, sulfur, it is involved in the formation of nodules and chlorophyll synthesis. It is a structural component of amino acids and enzymes, many important enzymes. Similarly, magnesium is involved in the regulation of enzymes and growth hormones. It assists in the photosynthesis and respiration. Molybdenum is involved in the enzymatic reaction of enzymatic reduction of nitrates into ammonia. It assists in conversion of inorganic phosphate to organic form. Similarly, zinc, it is involved in the production of growth hormones and chlorophyll. It actively involved in the respiration and carbohydrate synthesis. Boron, it is involved in the production of growth hormones and chlorophyll, and it participates in the respiration and carbohydrate synthesis. Similarly, copper, it is involved in the photosynthesis and respiration. Uh, Similarly, in the organic compound, carbon sources are used as sucrose is mostly used, but sometimes glucose, fructose, and other uh, uh, saccharides are used. In vitamins, they are mostly inositol is used, sometimes thymine, and some other adenines and other forms are used. So inositol is basically part of B complex, and it is uh, in phosphate form. It is a part of cell membrane organelles, and actually it is not essential growth hormone, but it is beneficial. Similarly, thymine, it is essential as a coenzyme for citric acid cycle. Similarly, adenine, it is a part of DNA and RNA. You know better. So the different hormone combinations are also used. Basically, four kind of plant growth regulators are used in tissue culture: auxins, cytokinins, gibberellins, and abscisic acid. But commonly, auxins and cytokines are only used, as you know that cytokinins they are mostly used as for callus formation, plant root growth and development, root rooting in the cutting, and in induction of adventitious embryogenesis. So various kind of auxins are used like IAA, IBA, NA, 2,4-D, and others. Similarly, cytokines, they enhance the adventitious root formation, it promotes cell division and growth, and it initiates axillary bud growth. Different cytokines are used as BA, 2IP, and many G18, kinetin, etc. Similarly, actually, gibberellins are not regularly used in the tissue culture, but it suppress the root formation and adventitious embryo formation. However, abscisic acid, it is preliminary a growth regulator, but it is more important for embryo zygotic and adventitious type of embryo formation. So in tissue culture experiments, we use the combination of auxin and cytokines for root and shoot development. And next slide, please. Yes. So for root formation, we used combination of higher amount of auxin and lower amount of cytokines. Similarly, for shoot development, we use higher amount of auxin and low amount of high amount of cytokinins and lower amount of auxins for shoot development. But if our target is to develop the callus, then we use equimolar concentration of auxin and cytokinins. You can see the in this graph, when the cytokinin will be present in lower amount and auxin will be present in higher amount, there will be root formation. But if the concentration will be decreased, then the formation of shoot will be increased, and in equimolar conditions, callus will be developed. Next slide, please. So any tissue culture experiments, there are few common steps. I think in your practical classes, you will understand it in better way. But I will brief only under sections like we can add different stocks for preparation of 1 liter of medium. Sometimes ready-made mediums are used. It is also nowadays it is also available in market in sachet pack form. So according to our requirement, we add our all the components in quantity, quantity of water like 1 liter. If we our target is 1 liter medium, then all the required salts we dissolve it according to their stocks, and then we prepared 1 liter medium. Then we add sugar accordingly, and we will set the pH. After setting the pH, we will add the agar, that is the solidifying medium, and then we will add the hormones. And then after adding hormone, we will make the volume according to our final volume according to our experiment. Finally, we will melt the media and placed it into sterile tubes. Then each tube will be leveled, and then a small piece of after solidification, the small piece of sterile explant will be placed with the help of a spatula in the this medium with under the laminar airflow, and then first we will use calcing medium that will contain equimolar concentration of oxygen and cytokinin, and then further we will change our our hormones. We will transfer it into the suiting medium as well, followed by rooting medium. So the first of all callus will be developed, and then after that after the development of callus, we will place it into the suiting medium. Then the suit will be formed, and after keeping each suit into a rooting medium, each suit will be developed into a separate plant. So next slide please. Yes. So the up to this, I think you will already know this thing in your class, but in next session we will see the practicality also. So I will discuss you about some my previous experiences, my previous experiments. So we developed the plant through the tissue culture techniques in Valeriana jatamasi. Actually, this Valeriana jatamasi is a medicinal plant, and its roots are commercially used for anxiety, insomnia, and many different diseases. So its roots have the market value of around 200 to 250 kg in dry form, and it is endangered at regional level. So with the help of tissue culture, we developed plants. You can see in the pictures, and then thousands of plants we generated. And next slide, please. You can see in the pictures, through the tissue culture, we generated thousands of plants, and then with the help of farmers, we not only placed these plants, tissue culture raised plant into the field, but we also supplied these plants to pharma companies also. Similarly, we also have an experience of developing tissue culture plants through the hydroponic medium. Next slide, please. Yes, this is a mass scale propagation of Hedychium spicatum. In our experiments, we also developed plants of thousands of plants of Hedychium spicatum. That's a medicinally important herb. And then after generating several thousand plants, we placed these plants into the farmer's field, and then supplied it to commercial purposes for pharma companies. Next slide. Yes, this is the experiment of our lab, Actually, we also developed the mass. Mass multiplication protocol for rhododendron species, various species. You can see that in Sikkim there are more than 36 species of rhododendron, and some of the species are very restricted to small area. So with the help of tissue culture, we generated thousands of plants of Rhododendron medonii, Rhododendron vipsanium, Rhododendron dalhousie, Rhododendron navius, and we then we raised tissue culture raised plant and placed it into the different conservation models like Gangtok Zoo, Plant Conservatory, Farm Bangla Wildlife Sanctuary, Kanchenjunga Biosphere Reserve, Kanchenjunga National Park, and many places. Next slide, please. Yes. Through tissue culture, we can generate tubers also, right? So in this slide, you can see that we developed the plants of Habenaria, and then we induced tuber formation in tissue culture raised plant in laboratory itself. So actually, Habenaria is edgeworthii is a medicinally important orchid species, and its tuber is important. So you can see that different steps we initiated tissue culture through seed-raised plant. After placing seeds into medium, then protocombs like bodies were developed, and then these protocombs like bodies were placed into the growth medium, liquid suspension medium, and then callus was found. With this callus, we induced shooting and then rooting. After rooting, we induced tubers also. So the medicinally important tuber, we generated this tuber in laboratory also without the help of soil. And interestingly, when we analyze the chemicals present in this orchid, we found that these orchids, the medicinal property of this orchids are relatively better than the widely occurring plants. Next slide, please. So there are other examples, successful protocol development of many medicinally important species. Next slide, please. Similarly, we can induce these. We can produce the secondary metabolites through the tissue culture medium also. So this is an example of tissue culture based suspension culture of Habenaria edgeworthii for secondary metabolite production. In previous slide, I have shown you the tuber formation. So when we analyze the medicinally important molecules, we found that the antioxidant and many important secondary metabolites were higher in tissue culture raised plants. And also when we applied some elicitors are basically molecules, those induce the stress-like conditions for plants, and in influence of this stress-like conditions, plant induce important metabolites. So induction of application of this kind of elicitors, we can induce the higher amount of accumulation of secondary metabolites. Next slide, please. Yes, sometime with the help of hairy root, we can accumulate higher amount of secondary metabolites in tissue culture raised plant. As you know, this technique is called hairy root culture. So in hairy root culture, it is basically type of rhizobium agrogenes mediated transformation of tissue. It contains root inducing plasmids that is called RI plasmid. So with the help of this RI plasmid, we can induce the accumulation of secondary metabolites. So these abnormal roots particularly have particularly they are easy to culture in the medium in tissue culture experiment. So this kind of neoplastic roots induced by the rhizobium agrogenes transformation have the higher growth, and within only few days we can get the full flask of roots in a very small time, and these roots will definitely have the higher amount of secondary metabolites. Next slide. So I will show you one example. We in our experiment we found that secondary metabolite production was higher in tissue culture raised hairy root in Picoraza curva. Actually, Picoraza curva is an endangered high value medicinal plant. So it contains important metabolites like picotin and picotoxin. After the transformation with different strains, in one strain you can see that we got around 15 time higher amount of picotin and around 10 times higher amount of picotoxin in tissue in hairy roots. Next slide, please. Similarly, there are other few examples of secondary metabolite production through the transformation mediated by agrobacterium rhizogenes. And next slide, please. Another application of biotechnology for conservation is genetic diversity characterization using the molecular markers. And these molecular markers are, you know, that essential tool for determining genetic variability and biodiversity at the molecule level. Next slide. For different kind of molecular markers are used in. So these molecular markers are extensively used for the conservation genetics also. These markers give the insight for the genetic process of the population, like mating system, gene flow, along with the information of past demographic events like bottleneck, genetic differentiation, or for the purpose of conservation measures. And it decline in the genetic variation use the ability of plant to adapt in the natural environment, and therefore decreased its in long-term survivability and sustenance. So the loss of genetic diversity can result in lower fitness of individuals and poor adaptability for the changing climatic conditions. So next slide, please. Different kind of molecular markers are used in the genetic diversity analysis. So we can categorize these molecules in these molecular markers into morphological, biochemical, and molecular markers. So next slide, please. Actually, the morphological markers, it is dependent upon the shape, size, structure-based characters. However, biochemical markers are used as a protein or enzyme profiling, and molecular markers, they are DNA-based marker, and they are most reliable marker because the biochemical and morphological markers can be influenced with the environmental conditions also, but these DNA-based molecular markers remain same throughout the life. So they can be considered as a factor for the evolution. So what are the basis for the genetic variation? Actually, genetic variation, before studying the genetic variation, we have to know the why this variation occurs. Actually, genetic variation is caused by mutation. That is, we can create entirely new allele in a population, or it may be recombination between the homologous chromosomes during the meiosis of cell division. So we analyze genetic diversity data in the form of allelic frequency. So along with few other parameters, but allele frequency is the relative frequency of allele. That is, allele is a variant of gene. So at a particular locus in a population. So allele frequency can be changed with in the influence of mutations, gene flow, genetic drift, or natural selection. What is mutation? You know that mutation is the alteration of any nucleotide sequence in a genome within an organism. It may be either due to the error in DNA replication process, or it may be caused by environmental process like radiation or any kind of stresses. Another is gene flow, that is transfer of genetic material from one population to another population within a period of time. If the rate of gene flow will be high, then the population will have an equivalent allele frequency among the population. Therefore, it may be considered as single effective population. Another factor we discussed that it is genetic drift. Genetic drift is a change in frequency of an existing variant of a allele within a population due to the random sampling of the organisms. Third and last one is natural selection. Natural selection is a process through which population of a living organism adapt to any changes. Natural selection of is a differential survival and reproduction of individuals due to the differences in its uh, phenotypes. So we can say that these factors are the forces for evolution in any organism. Next slide. Yes, this is an example of morphological and biochemical marker. Yes, this is one of our study in which we analyze the genetic diversity based on morphological markers like seed colors, seed length, width, weight, etc., and with uh, biochemical markers that was like protein profiling with the help of SDS page. So next slide, please. So in our study, we collected the different land races of rice of Uttarakhand, and then after SDS profiling, we found that these varieties are consist of 4 types of germplasms, and we targeted only 8 proteins, and on the basis of their presence and absence basis. So the some slow growing land races or less preferred land races, they may have some unique specific protein profiling. So on the basis of this exercise, we with the help of SDS page, we characterized 48 type of rice varieties of Uttarakhand. Next slide, please. This is just an example of genetic diversity analysis with the help of SDS page. Similarly, we can analyze the genetic diversity with the help of isozymes also. This is one example. In Hedychium spicatum, we analyze the genetic diversity with the help of isozymes, and among the 12 population of Hedychium spicatum is medicinal plant. We characterized and we found that the one population that is presented as H8 has the maximum genetic diversity and can be used as a genetic pool for ex situ conservation and variety improvement. However, some population like S2 and H11, you can see that these populations have very less genetic diversity. They have threat of extinction, and these populations need immediate attention for conservation. Next slide, please. So, in this way, PCR-based molecular markers can be developed for genetic diversity assessment in very fine mode. So, for genetic diversity analysis, we have to first isolate the DNA. Then, after PCR amplification with the help of optimum and suitable primers, then we analyze the genetic diversity, and further This genetic diversity, this profiling is analyzed for diversity characterization, evolutionary studies, elite selection, fingerprinting, or any forensic science related studies. Next slide, please. This is one example of genetic diversity analysis with the help of ISSR kind of primer, ISSR marker. So you can see that in 16 population of a medicinal plant, that is Hedychium spicatum, we analyze the genetic diversity parameters, and then
So you are not audible.
So everyone kindly wait. There's some network problem at our resource person's end.
मैम शायद सबका डिस्कनेक्ट हो गया है।
हाँ